Hi everyone, please ignore the birds chirping, but if I do this recording inside, I have my daughter's annoying dog. What we're going to do today is complete half of Unit 3 PowerPoint 2. This one is called Unit 3 PowerPoint 2A, and there's another one called B. So we'll finish off innate specific immune system, and we'll also cover hypersensitivities and immunity on the second part. Please take a minute to look over your outlines and also to download Respondus, because that's going to be necessary for taking exams in this class, as well as starting to work on your extra credit. It's going to be the questions at the end of your outline like it's always been before. Complete them. You can email them to me, but I'll talk more about that a little bit later. So last time we stopped off talking about interferon. And interferon essentially is something that is already inside of cells to protect them and give them resistance against viruses. You have a virus that comes in, attacks a particular cell, and that turns on interferon production. Once it turns it on, that original cell is actually going to die, but then the interferon will attach to the surface of the other neighboring cells and protect it. What it essentially does, or its pathogenesis, is that it stops transcription, or maybe I'll say it stops viral transcription, and that is the pathogenesis of how interferon works. Moving forward, we stopped the class last time to talk a little bit about complement, and I had mentioned to you that there's two different kinds. There's the one that is non-specific and the one that is specific. So today, we're only going to mention for now the non-specific ones in this lecture, and this one works very simple. It is released by the liver. It is in fact a protein, and it causes something known as a MAC attack, which is, as I mentioned last time, a membrane attack complex. Sorry, my mouth and hand cannot keep up with one another. And once it attaches to the cell or the bad guy that is causing the infection, it's going to pop it. Once you pop the membrane, it causes full lysis or the bad guy to pop. This is done in a non-specific manner. Here you go, membrane attack complex, and causes the lysis. Things that occur when complement actually happens are, hold on, let's get to the pointer. Things that occur are essentially the inflammation. So inflammation we'll discuss when we do the steps of inflammation. There are 4 of them. And it also causes, most importantly, I want you to all think about cytolysis, which is again a MAC attack, and then it lyses the bad guy. It does that by popping the membrane, which then causes everything to leak out and it dies. Now we need to speak a tiny bit about different types of cells in our body that are already there in our immune system to protect us. These are in fact our leukocytes, or white blood cells. We have many different ones. There are dendritic cells. These guys are what's called anchored. So every area in our body has dendritic cells. And the ones that are in your leg help fight an infection in that area. The ones that are in your arm help fight infection in that area. They don't move throughout the blood. They just actually stay and are specifically anchored. This picture right here is some kind of host cell. So we can say, I don't know, this is lungs of a patient. Doesn't really matter what it is. It's just some type of host cell. And there are different things that act as flags on the surface. Here's a handful of them, or 4 of them. These are things that in our body detect when a non-self cell or something is trying to attack us and cause an infection. Bacteria have LPS, specifically gram-negative bacteria. So if our body detects that, or something like ppt2, these are all things that don't normally exist in our own human eukaryotic cells. So what it's going to do is then turn on the immune system to help fight off infection. When you take a look at this picture that I managed to steal from McGraw-Hill, the ones that we're going to be focusing on are erythrocytes, or red blood cells. Those aren't too important at this part of the conversation. We have platelets. Their job is to act as clotting factors. So if you were to get a cut, it will help clot the blood so you don't bleed out. And then here are the 3 main leukocytes that we're going to talk about. Information you need to know about all of them are neutrophils, are the most abundant. That means they're found in the highest numbers. They float around in the bloodstream, and unlike dendritic cells that are anchored, these guys rush to the area in the highest numbers when there's, say you get stuck by a poker as you're hiking, or you cut yourself, or anything occurs. Then you have your eosinophils. These guys are most effective against helminths, so VS is versus helminths. And then you have your basophils. These guys act to release all kinds of cytokines, and a cytokine is a chemical signal that will allow your body to know, hey, I have to turn on the immune response, something is wrong. The only type of cytokine that we're going to talk about, since most of you are already familiar with it, are histamine. Lastly, here you can see dendritic cells. These are again anchored. Macrophages and phagocytes. This is what I described in the class as doing phagocytosis, which is the Pac-Man. And then we have our B and T cells. These guys are going to be part of the specific adaptive immune system and come in the second part of Unit 3 PowerPoint 2 discussion. T cells originate from, let's see, do this in a different color so you can see it easier. T cells come from the thymus, and B cells come from or originate in the bone marrow. That will be important to know later. So we have these white blood cells. They're in the tissues versus in the blood. The ones that are in the tissues are the ones that are anchored. So I'm just going to write DC, which is short for dendritic cells. And in the blood, we have the rest of them that are moving around. Some information about all of these cells is, as I mentioned before, eosinophils is best at killing off helminthic infections. They do something known as extracellular killing. So remember, a helminth is something like Billy the tapeworm that's in your GI tract. These guys are big. They could be meters long. So eosinophils are tiny. What they do is beat away and chip chop at it till they can break it down. They don't work very well, and eosinophils are by far the least effective leukocyte that we happen to have. Again, the neutrophils remain in the blood. These guys completely move around, and they are the most abundant. So here's a breakdown of putting them all together. The phagocytes cause phagocytosis. I want you to think of the Pac-Man as they go around chomping down the bad guy, which could be, say, bacteria trying to cause infections. We have dendritic cells. These are again anchored. Oops. Well, that's not going well. Hold on a second here. Okay. So dendritic cells are anchored. Neutrophils are moving around in the blood. And please know that every cell in the body has the ability to be a macrophage. All of our cells protect us to some point. You can take a look at these. So here again, most effective, most abundant. So read over this information on this slide to kind of give you a little bit more information about leukocytes. So this is a review slide that I don't necessarily have to bring up again, but what you're looking at is here you have a person who got, I don't want to say stabbed, but who got some type of splinter. Notice the splinter broke the blood, which means it's now entering the bloodstream. As we've mentioned in class before, when something enters the bloodstream, that essentially means infection is coming. So we poke into the bloodstream, which then automatically turns on basophils. Basophils release this histamine right here, which is how the body knows to react. In that particular area, bacteria, which are these little green tic-tac looking things, has been permitted into the bloodstream. Neutrophils, most abundant, come flying into the area. And then it's also important that you realize platelets are here. Remember, those are clotting factors so that you don't bleed out of this place where you've actually managed to break the skin. The 4 steps to the inflammatory response, which we did do in class, is erythema. This is redness. Pain is anything that ends in algia. Edema is basically swelling. And pyrexia is heat. If you touch a sprained ankle or where there's an injury, the skin will feel warm. Why is that? Because everything is working, as you see in this picture, rushing to the area. The blood is rushing to the area. The redness is from the bruising that you're actually going to have. So one last thing, or actually I guess there are two. We talked about interferon. Interferon is a completely non-specific immune response. It just happens. And so is one other one, which I really enjoy talking about this one because I think it's interesting. It's known as natural killer cells. And there are pictures up top right here and pictures on the bottom. These are essentially the same from different places. So I'm going to verbally tell you how this works, and then you'll understand why natural killer cells are known as the vigilantes of the immune system. A natural killer cell is already floating around in the bloodstream. When it encounters an abnormal cell, it actually attaches itself to it. Once it attaches itself to it, here, let's get a different color here. The Golgi of the natural killer cell, excuse me, aligns itself with the bad guy. It releases something called perforin, which then lyses the abnormal bad guy. So perforin is a very specific chemical found in the body that causes a MAC attack, which is again membrane attack complex. Please understand these are floating around in the tissues, also found in our lymph nodes. The one issue that it picks up on is if proteins flagged on our cells do not match the proteins flagged on abnormal cells, which is how it knows that there's a problem. Keep in mind, if the natural killer cells are programmed incorrectly, it does take an attempt at killing off our own healthy cells, which is similar to an autoimmune disease like we will talk about later. So what happens when all of these things are occurring? Well, if you think back to the picture about the inflammatory response, there is damage to the tissue. And when that happens, histamine is released because that turns on the inflammatory response. The blood vessels become dilated to let the blood flow more into the area. Why does that happen? Because again, what you want is for the neutrophils that are really abundant and good at helping us fight off infections. You want those rushing to the area. That area is going to become red erythema, swollen edema, warm pyrexia, and some type of painful. There is also clot formation occurring. This is due to the platelets. And then, of course, neutrophils are rushing to the area, and eventually, in due time, there will in fact be tissue repair of where the injury took place. So now we're going to bridge the gap a little bit between non-specific and specific. We've talked about physical barriers. The number one is your skin. We've talked a little bit about the leukocytes, which are neutrophils abundant, eosinophils, which are the ones that fight off helminthic infections. We've talked about natural killer cells and complement and interferon, which are antivirus infected cells. We've talked about fever, which is one of the steps of the inflammatory response. And now we have to look at how does non-specific eventually lead to your specific immune system being turned on. This is a picture right here of a dendrite. Remember, those are the anchored leukocytes or white blood cells in the area. What these guys do, if there's not enough response, will turn on our specific immune system. So to talk about specific immune system, this particular picture puts the whole thing together. So I'm going to go through this entire chart, and then we're going to break it down one at a time. First thing that occurs is there is an antigen. An antigen, in another name, is something called a non-self cell. This is something you've come in contact with if you have allergies, pollen, for example. If you get an E. coli infection from eating a bad taco, that could be the antigen. Anything that is non-self, doesn't belong, and turns on the immune system. It then activates an APC or a macrophage. Let me write on the side here, APC stands for antigen presenting cell. This is going to take the antigen that's coming in, act as a macrophage, which is your Pac-Man, and then present it on the surface. It's almost a flag saying, hey body, something is trying to attack me, and it's important that I fight this off. So notice how there are two sides. There's one side here and one side here. We'll discuss why later, but so far we're just working our way down the middle. Next comes a T helper cell that comes into the play. A helper cell assists with both sides of the immune system. The both sides are related to B cells and T cells because they fight off different types of infections in the body. The immune system is presented as if it's two completely separate systems for the sake of teaching it, but truthfully it is one big system that is working all the time. Maybe I'll back up and give you a little information here and say B cells, which are the B lymphocytes that originate in the bone marrow, is also known as humoral or antibody-mediated immunity. These are best at fighting off bacterial infections. On the other side here, we have our T cells. These are the T lymphocytes, and this side of, or this right side of the immune system is known as cell-mediated. This portion is best at fighting off viral infections and helping our body combat things such as cancer, which you'll see at some point later in the semester is more related to viruses than bacterial infections. But notice how we're back to the T helper cell here. It helps on both sides. A T helper cell, I want you to think of this as an assistant. This guy works at all times, no matter what type of infection you have, bacterial, viral, or something in between. The T helper cell will eventually take its time to attach to the antigen presenting cell. Between these two, there is a very specific, so to connect these two, there is a very specific receptor, and this is known as CD4. Every time the antigen presenter cell works with the helper T cell, CD4 is one of the two receptors that works. You're going to need to know both of them. There's CD4, and this attaches the antigen presenter cell and the T helper cell. The second receptor we're going to have to know about works only on the T cell side, so cell mediated, and this attaches, this is known as CD8, and this is going to attach at the cytotoxic T cell. Okay, back to the middle here. So now you have CD4 is the receptor that's attaching the antigen presenter cell and the T helper. At this point in time, the T helper is the smart one, and it will recognize, is this bacterial? Because if it is, then we're going to go this side. If it is a viral infection, then it tends to go more on this side. Remember a few minutes ago I said this entire system is working together, which in fact it is, but the other thing to consider is one side works a little bit more strongly, I'll say, depending on the type of infection that you have. So let's start with the T cell side right here first. So you have your T cell side, and what occurs here is there's what's known as a proliferation of T cells. What does that mean? Proliferation is a growth of. So a whole lot of more T cells grow because you can't have one single cell combating an infection. Once that occurs, the cytotoxic cell, this guy right here, is where CD8 is going to come into play. That's the second receptor I had mentioned a moment ago. And then a whole lot of cytotoxic, it's TC for short, cytotoxic T cells are floating around now in the bloodstream, working along with the non-specific things such as neutrophils, natural killer cells, interferon, absolutely everything else in the body to help create an immune response. Now, at the same time, while this is happening, memory T cells are also being made. This is, remember, for some type of viral infection. At the end of all of this, there's one last thing that's not on this chart, and that's a T suppressor cell. A suppressor, here I'll write it out for you, CUPRESSOR. A suppressor cell, when you suppress something, you stop it. So when there's enough of an immune response, the infective pathogen has been killed off, the TS is activated, or else it's going to start killing off your healthy cells, which we don't want. That is the T lymphocyte side. Now let's go back to the other side. We're back here again in the T helper. It's going to stimulate B cell response, assuming that we are combating some type of bacterial infection. Same thing happens as on the other side. One of the two aspects are memory B cells are proliferated or made. And then something unique occurs. B cells make plasma cells. Plasma cells have one job, and that is to secrete antibodies. This doesn't happen with T cells. Antibodies are only related to B cells. Take a look at the middle of this picture. I'm going to change the color here. So a quick review, you have an antigen. It makes an antigen presenting cell, which I'll show you on the next slide. What holds this all together is the CD4 receptor. Then the T helper cell decides, are we going to focus on the T cell side because it's viral, or the B cell side because it's bacterial? And then both sides make memory cells. And then the B cell side makes plasma cells that secrete antibodies. And the T cell side makes T cytotoxic cells. What is the name of the receptor that does this or helps bind it all together? It's the CD8, which is the second one I listed earlier. Lastly, I want to point something out to you. Up top here, it says first exposure to antigen. We're going down the line. And then notice right here in the middle, it says antigen second exposure. Let's say you get a second E. coli infection. At this point, it's a whole lot faster because your body knows what's happening. So immediately, it's going to refer to memory B and T cells and turn on the immune response right away. So this is a lot faster of a process when it's the second exposure as opposed to the first time that your body acknowledges it. So now let's do this one at a time. Remember that, as we talked about before, bacteria have all kinds of things such as, LPS, it has peptidoglycan, it has potentially a flagellum. These are all things that alert your body, hey, something's going on, we're being attacked, and it turns on the immune system. So we're looking at the first part of the chart right here. You have your antigen coming in. For now, we're going to say this is E. coli, since that's something you're all familiar hearing with. And now we have our antigen present. Oops, sorry about that. We have our antigen presenter cells. So what happens? The antigen will engulf it, which means it will eat or take in this E. coli. And then it's going to put little pieces of that E. coli on the surface like a flag. And it's doing that to warn the rest of the body to start up that immune response. So our B and T cells come into play. I've already mentioned this. The T cells are originating and processed in the thymus, whereas the B cells are in the bone marrow. These are found tightly packed into our lymph nodes, which help protect us. The lymphatic system is traditionally taught together with the immune system. I'm not going to go into detail with it, but I will mention that this works in response to when there's inflammation in the body and with the circulatory system, which carries the blood. And there are certain areas that you have lymph nodes that are very prominent. When you have a sore throat, if you touch the side of your throat, it's prominent. If you have a high fever and you touch under your armpits or on the side of the groin area, you'll notice that there's pain in that area. These are your essential lymph nodes that you are feeling because they're swollen. Okay, so back to the beginning. Here's the picture. You have antigen first exposure. Again, we are saying all along that this guy is E. coli. I think it makes it simpler since you're used to seeing it. That's our first exposure. Then it comes in here to the antigen presenting cell. So here is your antigen presenting cell. These guys are all macrophages. What does that mean? They have the ability to do the Pac-Man thing, engulf and then take stuff out. So when you have antigen presenting cell eventually attaching to the T helper, right over here, right, this guy right here. Here we go. This is your E. coli. It gets engulfed into the antigen presenting cell and broken down. And then it gets flagged onto the surface to help the rest of the body know something's going on. And this right here is the T helper cell. Remember, this is the assistant to help this entire process go. So, excuse me, there is a receptor here that attaches the APC and the T helper cell. And again, this is CD4. You need to know what CD4 and CD8 attach to. So please make sure you remember that. So now you have your T helper cells. So what happens at that point, if we are talking about T cells. The next steps are, don't worry so much about this right now, the next steps are the T cytotoxic, I'll write that word out for you. C cytotoxic cells are proliferated, or many of them are made, and T memory cells are made. And once all the process is done, the T suppressor comes in to stop it off. It is that simple. So here the whole thing is put together. Cell mediated is again related to T cells. So you start here, you have the APC, you have the bad guy coming in, and the T helper gets turned on. CD4 is the receptor that holds the two together. After that happens, the T cytotoxic cells, also known as T effector cells, they have 500 names, get turned on, and then we make a whole lot of them. This is the proliferation. Why is cytokine written right here? Because that is, we're just going to say histamine. That's the signal that is picked up when a non-self cell enters the body. Besides the T helper or T cytotoxic cells being made and proliferated, remember there are also many memory T cells made. At that point, cytotoxic T cells, as I mentioned, cause a MAC attack. So similar to natural killer cells, it releases perforin, which pops off the bad guy. And again, these are most effective against viruses and cancers because this is the T cell side. Then on the other aspect, we have humoral immunity or antibody mediated. These are your B cells, and they work very similar with very few differences. Here you have your antigen. Ooh, that's not going to show up. Hold on a second. Here you have your antigen presenting cell. This is your antigen coming in, breaks it down right here, and puts them on the surface, kind of like we've talked about already. This is the receptor holding the T helper cell and the APC together. So this is your CD4 receptor. Afterwards, we're going to go up here, lots of B cells are made. And the second thing that occurs, where is it? I'm going to write it down here for you. So first, a whole lot of B cells are proliferated. And next, B memory cells are made. And then plasma cells are made. Plasma cells have one single job, and that is they secrete antibodies. Why do they do that? Because one of the things that B cells have that's unique are, in fact, antibodies. The T cells do not secrete these, and B cells are best at fighting off bacterial infections. So putting this picture together, you have your E. coli coming in, you have your antigen presenter cell, here's your CD4 receptor, and now the T helper cell picked up this is bacterial, so it's heading in that direction. What happens is there's a proliferation of B cells, so many of them are made, along with memory cells and plasma cells that have one job, which is to secrete antibodies. So if you take a look at these few slides together, it completely explains this one picture right here. If you're able to put the last 4 slides into explaining this one chart, you're in very good shape. Please remember, second exposure means you have come in contact with the same bacteria or something a second time. Now, it doesn't have to go through this figure out which side it is. Right away, your body recognizes it, and it's going to turn on either B cells if it's bacterial or T cells if it's viral. Please watch this several times to make sure you understand it. I'm able to speak a little faster because you have the recording right in front of you to go over as many times as you'd like. See you next time for part B.